third-year medical student in University College Dublin. I completed this project along with Dr. Lambert and our collaborators. The goal of our project was a gene sequencing survey of Borrelia species in Irish ticks. And so our primary outcomes then were the prevalence of Borrelia infection, and then secondly, the specific species and the strains of Borrelia that were infecting Irish ticks. The Ixodes ricinatus tick is the principal vector of Lyme Borrelia in Ireland and the rest of Europe, really. It has a widening distribution in recent years. We now see it in Central Europe at higher altitudes and at wider latitudes as well. And this has been attributed to a combination of factors, among them climate change, changes in land use, and also the distribution of host organisms like cattle, like sheep, like deer. We see a diversity of Borrelia strains and species infecting ticks in Europe. In Ireland, in a previous study, three species were identified, among them Borrelia burgdorferi, Borrelia guerini, and Borrelia vasilani. Will someone correct me? Vasilani. Okay. So, with this study, we selected 6 representative sampling locations: Glendalough, Kilmacthomas, Killarney, Portumna, Clifton, Glenveigh. Ticks were collected using a flagging method, which involves using a white cloth to brush vegetation, which will collect the ticks, and the ticks can then be extracted from that. So in each of our sampling locations, we collected 50 ticks. These ticks were then disinfected with ethanol, 80% ethanol, dried in filter paper, and sent to a diagnostic lab in the US and Connecticut. Analysis of the ticks was conducted using a method of nested PCR. So this is a modification of classical PCR, where we have a first round of the traditional PCR followed by a second round of nested PCR. So this allows more cycles of PCR, so we have a greater amount of product and also hopefully greater sensitivity and specificity. And then the product of the second round nested PCR can then be used for Sanger sequencing, so we can have determine the specific base pair sequences of the strains of Borrelia and then compare this to a public repository of genetic data like GenBank. So then when we looked at the results, the 3 main strains that were identified, the 3 main Borrelia groups, were Borrelia guerini, Borrelia valesiani, and we also found a relapsing fever Borrelia, Borrelia miyamotoi, and this is the first time this species has been identified in Ireland. It was first identified in Japan in around 1995. Since then, it's had a higher profile, and we've also been identified in North America. It's been identified in England. And in addition to specifically identifying species, it's also important that we were able to identify the prevalence of these different strains. So we found overall the prevalence among the sample ticks was 5% were positive for Borrelia species. The most common Borrelia species identified was Borrelia guerini. This is followed by Borrelia valesiani, and then in addition, we identified 2 different strains of Borrelia guerini among the collected ticks. We had 2 among our sample where they were Borrelia guerini Bernie strain, as well as Borrelia guerini Vir strain. And in addition, we have Borrelia miyamotoi, a relapsing fever species. So then what can we draw from this? Well, when we see, looking at our prevalence, it's in line with previous studies. We have around a 2 to 12% range in prevalence. Previous studies have had a range from anywhere from 2% to 26%, depending on the sampling location. It's also important to note the specifics of the strains that were identified, because what we do know from previous studies is that when we're looking at the design of testing kits, the commercial testing kits that are available, they do show some variability in their sensitivity and specificity when accounting for specific strains. So a lot of the European test kits are calibrated for Borrelia burgdorferi, Borrelia garini, but there's some of the other strains like Borrelia valenciani, which may not be as well identified on classic antigen testing. So what we will hope is that we can endeavor to future research to actually determine how effective the current tests are and how effective any potential tests are at identifying some of these other strains which are prevalent in Ireland as well as other European countries. Another point to note is the presence of Borrelia miyamotoi. Relapsing fever has become more of a high-profile condition identified in pathogenic ticks. So future research should hopefully reinforce the findings of this study. And in the future, we can hope that we can have broader sampling to identify potentially a greater diversity of strains among Irish ticks. And this will help in public health strategies in Ireland to identify which are the highest risk areas in Ireland for tick infection and how best we can implement public health planning and diagnostic guidance for clinicians as well as future ecological surveys. Thanks very much.
Do you have questions? And do you know if there was a similar studies on the other side of the Irish Sea in Great Britain, or?
The generally similar species are found in Ireland. There hasn't been very many recent gene sequencing surveys done. So the last we had in Ireland was really in the 90s, so that we kind of need more up-to-date surveillance data. But I'm not sure specifically how this compares to, say, UK data. Probably a lower prevalence than we see in Scotland and parts of England, but still a prevalence that's worth considering.
Question, yes.
Do we have any? Not sure specifically. Jack, would you?
Just a question about sera. Right. The next part of the study that we'd like to do, obviously, is to just because they're in ticks doesn't necessarily mean they're in humans. So the next step would be so we haven't done the testing of the humans yet. The next thing is to make sure that the humans are infected with the ticks the Borrelia species that we identified in the tick study. So that will be the next step. So that's the next challenge. So maybe next year we'll be able to present that data.
Yes, a question here. Yeah.
Are you planning to do any other pathogen beyond Borrelia on those days?
The UCD Vet School just published in the Irish Veterinary Journal. They just come out about 2 weeks ago, and they did a review of and some pilots of what the small animals and wildlife and also domestic animals are infected with. And they're infected with, no surprise, Borrelia, Babesia, and Anaplasma. So these are the three predominant ticks that are in the wildlife. There seems to be no recognition. There's good recognition in veterinary, kind of scientists of these pathogens, but there's no recognition in the medical community. So I think the next step is if we could actually start working on that, that would actually raise the awareness of co-infections.
Okay, other question? No? Well, thank you very much.